वेलकम बैक अगेन डियर स्टूडेंट्स। आज हम बात करने जा रहे हैं किंगडम फंजे की। ओके, सो सम इम्पॉर्टेंट कैरेक्टर्स ऑफ किंगडम फंजे और यू कैन से डायग्नोस्टिक फीचर्स ऑफ किंगडम फंजे. The word fungi, it is derived from the Latin word. Okay, it is a Latin word. Fungi is a Latin word which means mushroom. And the diagnostic feature of the organisms belonging to fungi is they are achlorophyllous, which means they don't possess chlorophyll. Heterotrophic, they are not capable of manufacturing their own food, and they are thallophytes. That means their bodies are not differentiated into root, stem, and leaves. The branch of biology that deals with the study of fungi, this is known as mycology. Right? Now, the general characters. First of all, we will talk about their distribution. Fungi are cosmopolitan, which means they occur almost everywhere. They can occur in air, water, and the example is here Saprolegenia. Okay, Saprolegenia, here you can see this is the body of Saprolegenia. They cause infection of the fish species, and they can also occur in, I mean, their major species they are found in soil. They usually prefer warm and humid climatic conditions. Some fungal species, they are parasitic. They live a parasitic mode of life. They infect plants, animals, and human beings. There are some species of fungi which prefer to grow on dung. They are specifically named as Cropophilus. Okay, so Cropophilus species are those which grow on dung. Now the structure and growth. The fungal species, they are thalloid. Thalloid means they don't possess roots, stems, and leaves. And except yeasts, which are unicellular, yeasts are unicellular. Their bodies are composed of just a single cell. All the fungi are filamentous. The rest fungi, they are filamentous. These filaments, they are long, slender thread-like structures and are known as hyphae. The filaments are known as hyphae. And a network of hyphae, if you have a network of hyphae here like this network of hyphae, this is known as mycelium. Now the structure of hyphae. The hyphae can be having two types of structures. The first one is synostic. Synostic condition means when the hyphae are aseptate and multinucleate. Aseptate multinucleate means here this is a hypha. There is no internal partitioning. And this hypha contains many nuclei. Okay, there is no internal partitioning and the hypha contains many nuclei. This is known as synostic condition, and synostic condition is specifically present in lower fungi, phycomycetes. Now the second condition is septate with uni, bi, or multinucleate condition, and this condition is present in ascomycetes, basidiomycetes, and deuteromycetes. Here you can see this is a hypha. It possesses septum at many places. There are many septa. Okay, and here each segment, it contains either one nucleus in every segment, or it may contain two in every segment, or sometimes it may contain many nuclei in every segment. Okay, this is the main difference. One condition is known as synostic condition, and second one is known as septate with uni, bi, or multinucleate condition. Here you can see this structure, it represents the mycelium. You have a network of hyphae. This represents the unicellular condition in yeasts, and this one here you can see this is the synostic condition. Synostic condition means there are no intervening septa, and the hyphae contains many nuclei. Here this is also the synostic condition. Okay, synostic hypha, there is no internal septation, but this one represents the septate condition. Here you can see you have septa at many places and each segment contains one nucleus. Now the septal pores. The septa contains small pore-like structures or openings which are known as septal pores, and depending upon the structure of septal pores, there are two types of septa. One known as simple septal pores, and another one is known as dolipores. First of all, simple septal pores. Simple septal pores are present in case of euascomycetes and order Uridinales of basidiomycetes. They are present in euascomycetes and the order Uridinales of basidiomycetes. जो Uridinales है which belongs to basidiomycetes, they specifically contain simple septal pores. Now what are simple septal pores? Simple septal pores means, here you can see there is no thickening. Okay, there is no thickening of the septal membrane here. It is simple. It simply possesses pore-like structure in the middle part. Now the dolipores. Dolipores are barrel-shaped pores with septal pore cap or parthenosome. They are present in basidiomycetes except the order Uridinales, because Uridinales possess simple septal pores. Okay, here you can see this is the dolipore septum. It is having a barrel-shaped pore because of the thickening of the septal membrane. Okay, and this possesses one more additional structure that is known as septal pore cap or parthenosome. This is the septal pore cap, this red structure. Now the cell wall of fungi. The main constituent of fungal cell wall is fungal cellulose. It is actually chitin constituted of acetyl glucosamine. Okay, it is actually constituted of chitin, which is made up of acetyl glucosamine units. So the cell wall of fungi, it is unique. It does not contain the cellulose which is present in plants. It contains chitin. Okay, or you can say fungal cellulose. Okay, or simply you can remember the cell wall of fungi, it is not composed of cellulose, it is composed of chitin. However, there are some exceptions. The oomycetes, their cell wall is composed of cellulose and glucan. Okay, the oomycetes, their cell wall is composed of cellulose and glucan. Now the internal structure of hyphae. First of all, the cytoplasm. Cytoplasm is colorless and vacuolated. There are many vacuoles in the cytoplasm here. It is colorless and vacuolated. The endoplasmic reticulum, mitochondria, Golgi apparatus, they are present because the cells are eukaryotic cells. They possess mitochondria. There are endoplasmic reticulum which are present. Okay, here this is the endoplasmic reticulum, Golgi apparatus. All these structures are present. They don't possess chlorophyll and chloroplasts. However, the chlorophyll and the chloroplasts, they are lacking. In addition to these structures, the glycogen and oil droplets, you can find them in the cytoplasm. They store the reserved food material. Okay, they store the food material inside the cytoplasm. Glycogen and the oil droplets, they are the reserve food material. Now their nucleus. The cells may be uni, bi, or multinucleate. Uni means they possess single nucleus. Bi means they possess two nuclei. Okay, this is also known as dikaryotic, or multinucleate, which means the individual cells possess many nuclei. Now the dikaryotic condition. Dikaryotic condition, it may be further of two types: homokaryotic or heterokaryotic. Homokaryotic means when both the nuclei in a single cell, they are of same type. Same type, it means homokaryotic. And when the two nuclei in a single cell, they are of different type, this is known as heterokaryotic. Different nuclei. So the cells may be uni, bi, or multinucleate. Binucleate condition is also known as dikaryotic condition, and the dikaryotic condition is further of two types: homokaryotic condition or heterokaryotic condition. In homokaryotic condition, the two nuclei within a cell, they are of same origin, they are of same type. Okay, they are identical to each other. And the Heterokaryotic condition means when the two nuclei they are of two different types. Now their motility. The higher fungi, including ascomycetes, basidiomycetes, and deuteromycetes, they are immotile. They don't possess any motile structures. Okay, they are immotile. Yaad rakhna. The higher fungi are immotile. The lower fungi, in like phycomycetes, they are motile. The reproductive bodies, the reproductive cells, and asexual spores in these phycomycetes, they are uni or biflagellate. Here you can see these are the zoospores which are formed in phycomycetes. They are biflagellate or uniflagellate. Okay, they are biflagellate or uniflagellate. Now the types of flagella. Depending upon whether the flagella they are smooth or they bear hair-like structures, there are two types of flagella. One is known as whiplash or acrodymatic flagella. Whiplash or acrodymatic flagella are those flagella which are smooth. Here you can see they don't possess any hair-like structures on their surface, while as tinsel or pantonymatic flagella are those that possess hair-like structures or hair-like mastigonemes on their surface. These are the hair-like mastigonemes here on the surface of the flagella. They are known as tinsel or pantonymatic. Okay, so this is all for today. In the next part, we are going to discuss the remaining details of the kingdom fungi. Till then, stay blessed and thank you all.